Okay, hello everyone. Today our topic is medicinal drugs. So we're gonna talk about, especially we're gonna talk about modern techniques of making drugs. So before we start about talking modern drug designs, I would like to say something about traditional drug development process that was using the chemists was using the until the second half of 20th century. So what is traditional drug method? It's the traditional drug design is part of the trial and error method. What is it? Trial and error method is a fundamental method that is characterized by repeated actions, repeated attempts until the success is happened or practicer stops this test. So the main features about talking traditional methods is that it can be labor intensive and time consuming because to find one molecule of needed one molecule, the chemists and the scientists have to work with tens of thousands of molecules, different molecules, and today, by the help of technologies and modern techniques, chemists with collaboration with data scientists produced some different methods, modern drug designs. So here is some examples of drug designs, modern drug designs: structure-based, computer-aided, QSAR-based drug designs. So it consists of some virtual screening and molecular modeling. So what is it? What does it have especially in the techniques and in the design? They have three-dimensional structures of molecule. It helps us to view the structure of molecule in a molecular level, so that we can see the more precise and more pure. So it have the NMR, X-ray, and crystallography techniques. So, and I would like to say something about the example of modern techniques when the 1890s and 1880s scientists find a cure for AIDS. In Russian we say it's SPID. So AIDS, to find out the cure for AIDS, the scientists worked with its molecule. So they used X-ray crystallography. So using using its sample, they worked out the shape of HIV process. So they find out that molecule could be discovered that could block its active site. Here is a diagram of the enzyme. So its active site and substrate. So you can see this diagram. So eventually the scientists find out that working with modern structure techniques, we can achieve the more drugs and cure for the disease. Actually, at the end, they couldn't win this fight against AIDS because they created its mutated versions. So now today, scientists working with finding the cure. So what's the main points of these modern structure techniques? Why it's so important for us? So computerized techniques based on theoretical chemistry methods and experimental data to predict molecular and biology properties. So as we said before, traditional methods are so time consuming and high cost. So by working with modern drug designs, we can achieve the result faster and more cost-efficient discovery with discovery method. So and also only certain needed molecules are produced without trials and errors. So here is my topic, and others will continue about talking the talk.
Now we will say about synthetic drugs, like synthetic cannabinoids, phenylamines, synthetic cationes, tryptamines, and piperazines, novel benzodiazepines. Synthetic drugs is different by their chemical structures from the substances that are trying to mimic, and the manufacturers of synthetic drugs change their chemical structures to try and stay ahead to the law. Some synthetic drugs can be marketed as legal, safe, and acceptable alternatives to drugs. However, this does not mean that they are legal or safe. And if we will say more broadly about some synthetic drugs, phenylamines refer to a class of substance with documented psychoactive and stimulant effects, and includes amphetamines. And also methamphetamines and the MDMA, all of which are controlled under the conditions 1971 year. And the next type of synthetic drugs, synthetic cationes, they are related to the parent compound cation, one of the psychoactive principles in HAD. And the cation derivatives are the beta keto analog of a corresponding phenylethylamine. And the group includes several substances that have been used as active pharmaceutical ingredients of medical products, like amphetamine. And the most commonly available cation sold on the reactional market was mifepristone and the methylone. These products are usually used as highly pure white or brown powders. And the next type of synthetic drugs is the novel benzodiazepines, and they represent a heterogeneous group of drugs that are increasing in number as new compounds are continuously developed, and they are being seen with increasing frequency in forensic and drug programs as drugs of abuse in recent years. Several compounds with significant differences in their duration of action, mechanism of action, and their structure are reviewed as examples of key subtypes among the novel benzodiazepines. These are 4-chlorodizepam, pentazepam, antizolam, fluprazolam, and pharmacodynamics and pharmacokinetics are reviewed, were known, as well as prevalence of use and the acute and the chronic toxicity reports. In the next topic,
Okay, next topic is drug delivery, and I'm not sure that the right definition for to our theme, but drug delivery refers to the technology utilized to present the drug to the desired site for drug release and absorption, or the subsequent transport of the active ingredients across the biological membranes to the site of action. And as you can see, here's a not very clear photo of the drug delivery system, but I can message after the presentation clear photo of this structure. So next is problems of drug delivery. As you can see, here the many definition, like the use of large-size materials in drug delivery poses major challenges, including in vivo instability, poor bioavailability, and solubility. Also, absorption in the body, issues with target specificity. So there is a kind of very simple and very simple problems in this theme. Also, here's a question about the most serious problem in the drug delivery system. So I found the very easy answer for this. The major challenge for any drug delivery system is compatibility and acceptability, because interaction of synthetic materials with human body cells is entirely different from biological one. So here's the main problems of drug delivery and the main thing that we need to know about the drug delivery. So that's all of our work in teamwork.